From individual fingers to our entire bodies, our muscles are moving all the time, every day. All muscle movements are caused by a contraction at the neuromuscular junction. The neuromuscular junction is a site where an electrical signal from a neuron can interact with a muscle cell. At this junction, a neural message can be translated into muscular contraction and movement. To send a message to a muscle cell, the motor neuron first receives a signal from its dendrites. Then the signal is passed along by the axon to the axon terminal and the neuromuscular junction. The axon terminal is at the very end of the axon, and it handles communication between the neuron and other cells. When the axon terminal receives the proper signal from the motor neuron, it's triggered to absorb sodium and calcium. Calcium then binds to the axon's vesicles, which release the neurotransmitter acetylcholine by fusing their membranes with the membrane of the axon in a process called exocytosis. Acetylcholine acts as a messenger by binding to receptors on the muscle fiber's sodium channels. By binding to the receptors, acetylcholine tells the channels to open and allows sodium to enter. Eventually, enough sodium enters that calcium can enter as well. Calcium is able to regulate muscle contraction by interacting with the proteins troponin and tropomyosin, which in turn interact with the proteins myosin and actin. Actin and myosin work together to generate the force behind a contraction. When ATP binds to it, myosin uses the energy released by ATP's loss of phosphate group to take a high-energy position that slides it forward along the actin. When the phosphate group then detaches from myosin, myosin slides back to its low-energy position, which pulls actin. But without outside intervention, myosin is unable to interact with actin. The myosin-actin binding site is blocked by tropomyosin, which is kept in position by troponin. However, calcium can remove this barrier. If there's enough calcium in the cell, it can bind to the troponin and change its shape, allowing myosin to move from its blocked position. Even for something as simple as blinking, this entire process takes place in split seconds whenever we move. Isn't that amazing?